Now, of course, proteins are not these static structures, but they're moving. And then, of course, when you compile with, and this is actually I think this is a 5 picosecond time snap of water, a membrane, and aquaporin in our kidneys and so forth. So protein stability. What's so important about protein stability? So you see protein instability maybe during breakfast, right? This is albumin. And this is actually protein denaturation that you're viewing in real time. Right? Your proteins are actually denaturing. So you don't want that to happen. So this is a simplified view of what happens if you heat a protein. If you get 100% folded, and then you approach as you increase in temperature and so forth, you get what they call an unfolded species or so forth. But in the meantime, in the middle of this, there's one of these strands and so forth. They're starting to move out, right? So they're starting to partially unfold. These are the problem proteins. These are problems, and these can exist if you just have one amino acid change in some cases. So, for instance, cystic fibrosis. Person that has cystic fibrosis, the most dominant change of cystic fibrosis in their defective protein is one amino acid change. They deleted one phenylalanine. F508, phenylalanine 508, it's deleted, and that causes the disease. One single amino acid out of the whole. So the problem you've run into, of course, is that you run into a situation where if this starts to accumulate, this is where you have this source of aggregation. And if you have a mutant protein, no longer do you have 100% folded. Maybe you've got 75% folded. But then it really gives you a larger population of this unfolded protein. So in a mutant protein, that if you have a mutation and so forth, you're spending a lot more time in this misfolded state. So here's an example of a transition. I'll have to make sure that, okay. So here's what you're seeing is this transition from one state to the other. This loop actually inserts into this groove here, and this goes from an active form to an inactive form. But just want you to point out the fact that these proteins are actually fibrulating, going back and forth between these two states. So proteins can take many different paths, right? They're synthesized from the ribosome, translated, partially folded form, and you can have this misfolding, and hopefully your body can clear those. You can have situations, unfortunately, if this stays around for a long period of time, if you have a mutation, then these proteins aggregate. You want to actually get to this point. So one of the things that we try and do in our lab is try and figure out ways of detecting this partially folded species and then making sure it acquires a fold rather than a misfold. So here's an example of these proteins that are actually in a folding intermediate. Here's the native protein as this folds, but sometimes the loops are hanging out here, and they swap with other proteins of the same species that are having the same problems. So you'll get these long aggregates and so forth. And these are, you can see that there's swapping domains between these two, swapping domains. So this is what aggregation is all about. They're very specific in aggregation. They're not just a glom. Here's an antitrypsin inhibitor. This is actually a anti-elastase. So one of the this circulates in our blood. It was called the alpha protein before, because we were doing paper electrophoresis. We were like in alpha bands, beta bands, gamma, and so forth. And this protein turned up in the alpha band. It actually inhibited trypsin, but it turns out that this protein inhibits elastase. So elastase is important for macrophages to get through the milieu and get into new, where you have bacterial invasions and so forth. This is where they actually pump out this protein elastase to chew up and enter into regions where bacteria are populating. But if you have a mutation in this, you can't inhibit your elastase and they go wild. And then you start to digest your lungs. And that's what happens with people that have this folding disease with emphysema. And lo and behold, what happens is that as this is transported in the liver, it goes from one organelle to another, there's a pH change, and there's some destabilization of a loop here. And lo and behold, what happens is that this loop sticks out here. Another one's having the same problem, but that little groove is still available for interaction. So what you can get here is actually aggregates, very specific aggregates, and then with electron microscopy you can actually see those aggregates. And this is actually an event that occurs in emphysema for genetically and this is only one amino acid change that can result in this. So what you'd want to do, and what we're trying to do, is actually trying to prevent that transition from occurring. Here's the other one that's amyloid fibers. This is these are the ones that actually accumulate in your brain when you have Alzheimer's and so forth. And they actually form nice little ribbons. Actually, they're not nice, but they form ribbons and regular structure and so forth. And it turns out what's really kind of annoying and actually problematic is that you can turn a lot of different folds into these aggregates, into these very specific amyloids. And that's what happens with, actually I don't have this here, beta amyloid, Alzheimer's, alpha-synuclein, Parkinson's, transthyretin, that's a neuropathy, cardiomyopathy, also prion protein, that fatal familial insomnia, that's the same, that happens, So when we look at disease, we have this manifestation. It impacts physiology, anatomy, cell viability, cell biology, and so forth. It has many different effects. That's what pleiotropic means. So what we do is actually try and figure out what the molecular basis of these folding diseases are. And usually we end up with a single amino acid change, and resulting in cystic fibrosis, sickle cell anemia, Parkinson's, and so forth. But once we understand this molecular impact on the macroscopic system, then we devise therapies. So what kind of therapies are we going to be talking about? So here's the interior of a cell, and this is where it just shows you how crowded this environment is. Now, in our cells, we've got huge amounts of concentration of proteins, but we don't get aggregation. Why is that? And this really can be a problem, because in the sense of going from 37 to 41 degrees in E. coli proteins, more than 400 E. coli proteins are destabilized if you were just paying attention to the free energy, the stabilization of these particular molecules. So that's a problem, right? So how does the cells actually prevent this from occurring? They have a class of proteins. When I was in graduate school, they had no idea what these things were. They called them heat shock proteins. So when you elevate your temperature and so forth, there's proteins called heat shock proteins that are elevated. It's one of the reasons why you kind of feel the first hot day of the year, right? You're just kind of like, oh boy, you're elevating your heat shock response and so forth. So these proteins actually prevent these aggregations from occurring, and there are all kinds. They're called heat shock proteins. And the heat shock protein 70, this is 70 kilodaltons. Heat shock 90, that's 90 kilodaltons. And there's large chaperones. There's sponge proteins. There's heat shock proteins in our eyes. So cataracts, of course, are protein aggregation. Right? So we have chaperones in our eyes that prevent protein aggregation. They're called small heat shock proteins, and these are just simply sponges that bind to aggregated proteins and prevent them from forming larger aggregates that reflect light. So, they scatter light. So the protein we use as a tool, as a detector, is called the chaperonin protein. This actually resides in the mitochondria. So the mitochondria, a lot of proteins actually are transported from the exterior into the mitochondria. Something like 1,500 proteins have to be transported, are synthesized out here, unfolded, refolded on this side. And so when you can see, one of the problems you have with the heat shock proteins and so forth, as you start to aggregate or start to build up aggregates out here, this interferes with your mitochondria function because you can't import those proteins into the mitochondria. So that's where some of these protein folding diseases come from, and actually our neurons are just absolutely chock full of mitochondria, right? If you look at a real-time firing of brain cells of neurons, they're just popping, right? Boo, 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 boo. It's a storm, literally a storm, even when you're sleeping. And that energy comes from the mitochondria. If you start to, and what happens with neurons is if they have defects in their transport, they're actually going to have defective mitochondria, and that's one of the reasons why the neurons actually start dying. So these proteins help fold proteins. Sometimes there's specific assembly proteins. There's chaperones. There's unfolding chaperones that actually unfold misfolded proteins and get them back on pathway. And then there's also disaggregation proteins that actually disaggregate. So these heat shock proteins are vital to the system. And the protein I work with is called the chaperone, and actually it can encapsulate proteins in a very nanostructured chamber and fold them and release them once they're folded and so forth. So these go back in a two-stroke cycle. But we actually use a form of the chaperone that's has the highest binding affinity. This is the form that actually exists in the cell, but we found that this protein, without ATP and so forth, has a very high affinity for hydrophobic proteins. And here's a cryo-electron microscopy view of just 4 subunits with a protein bound. So this is partially folded. These are hydrophobic residues. These are oily residues. So they capture these proteins as they're unfolded or partially folded, and these hydrophobic patches actually bind here. So what we devised is a way of detecting whether or not we can cure or reverse misfolding using this detection system. So this is the technology platform for detecting misfolding in pre-aggregates. Here's our disease misfolding forms, right? This is what we want to end up with in this folded form, but we run into this problem if we have a mutation.